Muy buenas tardes. Agradecemos mucho el interés y que nos acompañen en este ciclo de conferencias de Universidades por la Ciencia, organizado por Fundación UNAM, el Colegio Nacional y un conjunto de instituciones de educación superior y de investigación en México y también en otros países. El consorcio de Universidades por la Ciencia tiene alrededor de 20 instituciones participando. El ciclo de conferencias presenta, que es la característica principal, investigaciones que están en desarrollo. Es una de las formas en que pues, la ciencia se va construyendo. El día de hoy tenemos una conferencia muy especial a cargo de la doctora Leslie Turner del Centro Milner de Evolución en la Universidad de Meade. La conferencia es sobre la parte de la generación de nuevas especies, de especiación, y esto es la base de la evolución, la base de cómo la vida, las diferentes formas se han ido desarrollando a lo largo de los miles de millones de años. ¿Cómo se forman las especies? Es una de las preguntas clave. Y esto ahora tiene la ventaja de poder ser investigado con mucha mayor resolución a través de los estudios de genética, de genómica, los cuales nos permiten investigar en donde este proceso se desarrolla. Y el trabajo que desarrolla la doctora Turner y su grupo es particularmente interesante y utiliza uno de nuestros organismos más emblemáticos, los ratones, que fueron, de hecho, pues vienen descendiendo de la extinción masiva que acabó con los dinosaurios, fueron de los únicos grupos de mamíferos que lograron pasar esta extinción masiva y que nos dan la diversidad de especies que tenemos, incluyendo a la parte de los homínidos y de los humanos. Entonces, el trabajo de investigación que realiza la doctora Turner y su grupo es particularmente interesante y esto nos va a permitir el entender cómo las diferentes especies han ido desarrollando. Y le agradecemos mucho. Thanks very much, Leslie, and welcome. We really appreciate and thanks for accepting the invitation. In this conference series, Universities for Science has been particularly successful and presents the research projects in progress. We characteristic of this conference cycle that we are presenting research as it is being done. Nos acompañan el licenciado Dionisio Meade, que es el presidente de Fundación UNAM, y la doctora Araxi Urrutia, que es del consorcio de Universidades por la Ciencia en la UNAM, y la doctora Leslie Turner y la licenciada Araceli Rodríguez, nuestra directora ejecutiva de Fundación UNAM. Entonces, nuevamente, pues a nombre de todos, les agradecemos muchísimo el interés. Esperemos que sea muy atrayente y muy importante esta conferencia. Y le paso con muchísimo gusto la palabra a nuestro presidente de Fundación UNAM, el licenciado Dionisio Meade.
Muchas gracias, Jaime. Muchas gracias a todos por acompañarnos. Muchas gracias, Leslie. Gracias, Araxi. Para nosotros es un motivo de singular privilegio participar en este consorcio y agradecer al Colegio Nacional y a la propia universidad y a la Universidad de las Américas de Puebla que hayan tenido esta iniciativa como una respuesta a los retos de la pandemia. La pandemia trajo consigo la dificultad de comunicarnos presencialmente, pero al mismo tiempo significó el desafío de tratar de encontrar nuevas vías de acercamiento, nuevas vías de contacto entre la investigación y la academia. Y derivado de esta iniciativa, pues se ha generado la operación de este consorcio que cada vez suma más universidades afines y que ya suman 20, nos decía ahorita Jaime, y que tienen la virtud en las presentaciones que nos hacen de hacerlas con investigaciones que están en curso. Esto nos permite compartir los desafíos de la ciencia y al mismo tiempo estar al día en los temas más sensibles, más avanzados de lo que está haciendo la investigación científica. Hemos dedicado este ciclo como homenaje a dos grandes personajes que tuvieron esta inquietud por la investigación científica y que hicieron grandes aportaciones a la humanidad. Me refiero desde luego al doctor Mario Molina, nuestro premio Nobel, y al doctor Guillermo Soberón, que fue rector de la universidad, y quienes con su vida dieron testimonio de los frutos que pueden derivar de esta investigación científica. Nosotros quisiéramos que muchos más jóvenes se sumaran como consecuencia de la presentación de estas investigaciones, a quererse sumar al esfuerzo del conocimiento, al esfuerzo de la investigación, y que pronto veamos en cada uno de ellos el fruto de esta vida dedicada a la investigación y a la ciencia. Las presentaciones que hemos tenido de los consorcios han permitido enlazar a universidades de muy distintas partes del mundo. Hoy tenemos a Leslie, que nos hace favor de acompañarnos desde la Universidad de Meade. Ya hemos tenido otras presentaciones de ahí mismo y hemos encontrado también que mientras que en las actividades presenciales las posibilidades de atención pues se centraban en decenas o centenas, en cambio las que estamos logrando a través de estas presentaciones pues se cuentan por miles. Se cuentan por miles y además por muchos puntos del planeta, de nuestro país, que están siguiendo la presentación de estas pláticas. Así que es de veras un motivo de mucha alegría, es un motivo de una gran motivación también para continuar con estas pláticas. Y hoy pues vamos a agradecer esta presentación sobre la evolución y sobre la evolución de los ratones. Y ahora caiga en la cuenta de que el cuadro que tengo detrás es de un gato. Espero que no se vaya a interpretar como una alusión en contra de los ratones. Eso fue una circunstancia de la que apenas ahorita me doy cuenta. Muchas gracias. Gracias, Leslie, por tu presentación. Y como decía Jaime, estos temas de la evolución y cómo se fueron generando las especies son sin duda unas de las que más interés suscitan y unas de las que pudiera interpretarse como más profundos misterios en la vida de las especies en nuestro planeta. Muchísimas gracias y encantados de escuchar tu presentación. Thank you very much, Leslie.
Thank you, Dr. Urrutia and Licenciado Dionisio Meade, por las palabras. So it is a great honor to have Dr. Leslie Turner giving this presentation today. She is a very great scientist and also a very dear colleague. So we appreciate very much that she's generously giving her time to join us today for the closing presentation of this webinar series of Universities for Science Consortium. So we started back in June, and we thought that what was happening would only last a couple of months. Here we are 7 months later. We're still sort of not back to normal, but we are very happy with the response to this seminar series. So to close, we're going to have a talk about speciation and what is it that we can learn about the origin of species from examining and studying wild mice. So Dr. Leslie Turner did an undergrad degree in biology at Barnard College, and she followed that by a master's in biological anthropology and anatomy at Duke University. And then she did her PhD at University of California, San Diego, followed by postdocs in Harvard and the Max Planck in evolutionary biology in Germany and the University of Wisconsin. Since 2016, she joined the University of Bath at the Milner Center for Evolution and the Department of Biology and Biochemistry, where she leads a lab looking at mice and examining what is it that changes in the genome to control speciation, but also what are the consequences at the genomic level that can lead to male sterility when you have hybrid mice coming from two different subspecies. So Leslie, thank you very much for joining us, and we look forward to your presentation. Thank you.
Thank you. Thank you very much for inviting me. And as I was saying before we got started here, that one of the few benefits of this pandemic is that at least we can have the opportunity to share science across long distances more than we used to in the past. So I've enjoyed watching already some of the talks in the series, and I'm glad to have the opportunity to share a bit about our science today. So let me share my screen here. Okay, so yeah, so I'm going to tell you about how we're learning about how speciation occurs by studying wild mice. And my motivation throughout my career has been to understand how we get the diversity of life that we see on the planet today in form and function, and how does that arise? And the process of speciation, so how all those different forms get created, has been a topic of interest for a very long time. Of course, in biology, and Darwin's most famous work, of course, was called The Origin of Species. However, Darwin really talked in Origin more about adaptation or change in species through time, and he didn't very much address the exact process of how one species splits into two. So, for example, what were the processes that caused the original finch that invaded the Galapagos to diverge into the variety of different finch species that we see today? So we're still interested in what is causing speciation. We're still an active area of research. And in particular, what I'm interested in are the genetic changes that underlie the speciation process. So if we want to start studying species, first we have to define what a species is. And I use the biological species concept, which is convenient for those of us who are trying to understand the genetics, because it defines species as groups of organisms which are reproductively isolated from other groups. And so by focusing on this characteristic of reproductive isolation, then we have a specific thing to look for when we're trying to understand speciation. We're trying to understand what causes reproductive isolation. And here is a cartoon version of speciation, where you have a group of mice that get split by some sort of geographic barrier, so that over time, because there's this barrier there, then different genetic changes accumulate in each of these subpopulations indicated here in red and blue, such that when they come back together, if this barrier goes away, then they can no longer freely exchange genes and breed and produce fit offspring. So what kind of barriers can cause those species to remain separate? There's barriers that happen before fertilization, pre-zygotic barriers. There's several different forms of those. And one example is shown here by these cichlid fish, which use color for mate choice. And so even though different species can be crossed in the laboratory and produce perfectly fit offspring, because they use this color for mate choice, they don't recognize each other as mates in the wild, and so they have this barrier that occurs before fertilization. There are also post-zygotic barriers that happen after fertilization, including hybrid inviability, hybrid sterility, and then ecological selection against hybrids. And this is one of my favorite hybrids, which is a zorse, which is a sterile hybrid between a zebra and a horse. And so I'm going to tell you today about our work on two different kinds of postzygotic isolation. One is looking at hybrid male sterility particularly, and here's some more fun examples of hybrids that are sterile. So the mule is sort of the classic example that everyone's familiar with, which is a sterile hybrid between a horse and a donkey. But then there's fun ones like ligers and donkeys. That one's pretty cute too. So there's lots of different kinds of hybrids. Unfortunately, the mouse hybrids are not as exciting to look at, but they're pretty useful to study. So I'll tell you about hybrid sterility, and then the other form I'm going to tell you about is changes in the gut microbiome. And there's been much less done on how the gut microbiome contributes to speciation, but there's one interesting case where gut bacteria are responsible for causing hybrid lethality if you cross two species of wasp in the genus Nisonia. And then I found another example of changes in the rumen microbiome in this hybrid here, which is between a deer and an elk. So that's less understood, but as you may be familiar, gut microbiome is a very active topic of research in biomedical research. So we're interested in seeing how that contributes to speciation as well. So I told you a bit about what the reproductive barriers are. So what are the traits that can cause speciation? But we also need to think about what are the genetic changes. How do those genetic changes arise? So here is the Dobzhansky-Muller model, and this is was proposed by Dobzhansky and Muller independently to explain how genetic changes can cause speciation. So this is a two-locus model, so it involves two different genes. And so if you start off here with the ancestral genotype being, A1, A1, B1, B1, then in one of the populations, a new allele arises in gene A, A2 here, and then goes to fixation. And in the other population, a new allele arises at gene B, B2, and goes to fixation. So that when a hybrid is produced, these two alleles, A2 and B2, have never before been in the same organism. So they can potentially not work together and have what's called a hybrid incompatibility, or Dobzhansky-Muller incompatibility. And so this is important because if you think about it from the perspective of just one gene, so say we were just thinking about the A gene here, then at some point, if you have a new genotype, then you had to have a heterozygous genotype here, this A1 to A2. And if the problem was caused just by this one gene, then this heterozygous genotype would have had reduced fitness. And so it's not plausible that the species would have gone through that heterozygous fitness valley. But if you consider it from this two-locus model, then that gets around that problem because these alleles didn't have to interact with each other when they were evolving. And so only now when they're in a hybrid organism for the first time are there problems caused. So if we want to understand genetics of speciation, at least postzygotic isolation, then we need to look for what are these Dobzhansky-Muller incompatibilities that are causing problems in hybrids. And so there have been two main approaches classically in speciation genetics. One is performing crosses in the laboratory, and this is limited to mostly to genetic model organisms, primarily Drosophila species. And so by performing laboratory crosses, then people have identified regions of the genome that are associated with these reproductive barriers. And the other main approach has been looking in natural zones of hybridization. So for example, many different species of heliconius butterflies form hybrid zones. And so looking at how genes move through natural hybrid zones to find regions of the genome that might contribute to speciation. And these approaches have been quite successful and have really advanced our understanding of speciation. And for example, there's more than 20 so-called speciation genes that have been identified in the laboratory, including one in mice, not by me. And then in nature, there have been many regions of the genome that have been identified that have reduced gene flow. So here's an example from mice here, from the hybrid zone that I work on between two subspecies of house mouse in Europe. So there's the Western European house mouse, Mus musculus domesticus, and then the Eastern European house mouse, Mus musculus musculus, that meet in this hybrid zone. And in this study, they showed that these are showing patterns of variation at 2 genes across the hybrid zone. The frequency of musculus type alleles. And so you can see here that there's a very drastic change as you move across the hybrid zone from things that are domesticus, so 0% musculus, to 100% musculus. And so these two have these particularly sharp clines. What we call it, the shape of the cline is very severe. And so that suggests that there's some reason why the musculus genotype doesn't do well in the domesticus background and vice versa. So those regions of the genome might contribute to reproductive isolation. So those are the two main approaches that have been done classically, but rarely are they done in the same organism. So most of the species that are studied in the laboratory, like most of the species of Drosophila that have been looked at so far, don't actually hybridize in nature. And then most of the species that have hybrid zones in nature don't have, or until recently, didn't have the genetic tools available to be able to map the regions associated with traits. So that's why house mice are actually a very good model for understanding speciation, because as I mentioned, there's a hybrid zone between 2 subspecies in Europe, and there's actually 3 subspecies of house mice. Mus musculus castaneus is an Asian subspecies, and all 3 of these can interbreed and do when they meet each other in nature, and they diverged in the last 500,000 years, so quite recently. And then, of course, because they're the wild relatives of lab mice, we have a large array of genetic and genomic tools that we can use to understand the genetics of reproductive isolation. And importantly for me, they're the premier model of understanding reproduction in mammals. So we know quite a bit about what's going on with their fertility, and as well their gut microbiome that I'm going to tell you about. And so here's the pictures of the 2 subspecies, and the only reason that I know that this one is domesticus and this one is musculus is because I chose the pictures that way. As you can see, they don't look very different from each other, so they're not quite as interesting to look at the hybrids as Azores or something, but you can tell them apart at the genetic level. And they do have some phenotypic differences. They're just not very obvious. So one of the reasons why, I mentioned that they're in the early stages of speciation, and why that's important is that if we blow up this figure here into a little bit more detail. So here I've drawn lines, and the little cross marks there are indicating different genetic changes that are arising, with those that might cause reproductive isolation are in color. And I've shown gene flow here as an arrow. And so when they first separate, they have full gene flow. And then the propensity to be able to exchange genes goes down over time until finally they accumulate enough changes so that they're fully reproductively isolated. And we are interested in the ones here shown in green asterisk. So the genetic changes that are causing reproductive isolation to happen. But once it is complete, there's still going to be additional changes that are accumulated in these different lineages, so that those could also cause problems when you cross them in the lab. And so unlike some other species, like for Drosophila melanogaster and simulans, which are far past this point of reproductive isolation, mice are in these early stages, so we can find these genetic changes that are currently reducing gene flow between the subspecies. So I mentioned that there's many different types of reproductive barriers, and I mentioned a couple in mice, but we don't know, we know that there's many different forms of barriers between them. So for example, some hybrid mice have higher parasite loads than pure subspecies. They have some evidence for assortative mating, meaning that females of one subspecies prefer males of the same subspecies and versus the other, but that's not super strong. Like I mentioned, there's reduced hybrid male fertility. There's also reduced hybrid female fertility. And so those have been quite well studied. And there's a previous study I'll tell you about looking at the gut microbiota being altered in the mice. So as I mentioned, I'm going to tell you about two of those today, hybrid male sterility and gut microbiome. So I'll tell you first about a genetic mapping study that we did previously of hybrid male sterility using first generation lab bred males. And then I'll tell you about how we developed these hybrid inbred strains to be able to have better power to understand the genetic speciation. And then I'll tell you about, these are hot off the presses, so very recent results that we're currently writing up on mapping gut microbiome traits and a cross between these inbred strains, and in mapping some more sterility traits of them. So I went to the Bavarian region of the hybrid zone back almost 10 or more than 10 years ago now, and caught mice in this little block, this little black box here in the hybrid zone. This was in collaboration with Bettina Haar. And so I caught mice in this 40-kilometer transect shown here. And this graph shows the percent of musculus alleles, so alleles that distinguish between the two subspecies as you go through the hybrid zone. And so you can see over here, they're purely domesticus, so about 0% musculus. And over here, they are fully musculus, and that's over a 40-kilometer distance. And so what we're interested in is what's going on in these, this very sharp change you see between the two subspecies. Why are they being kept apart? Why aren't they just joining back together into one species now that they've met here after being separated? And so we genotyped all these mice, and then the ones here that are have black dots, we brought back to the Max Planck for evolutionary biology to breed. And we were interested in figuring out what degree of hybrid of male sterility there was in the hybrid zone mice. So previously, people had crossed the two subspecies in the laboratory and shown that there's hybrid sterility, but there hadn't been a good documentation of what level of sterility there was in wild hybrids. So we brought back about 50 males and 50 females to the lab, and we bred them so that we could get as close as possible to a wild hybrid, but born and raised in a common environment. So we took males and females from that were captured in the same barn or the same stable and bred those, and then looked at their first generation male offspring. And so we looked at a variety of different fertility traits, so testis weight, sperm count, sperm motility, etc. And here's some examples. So mice have this characteristic hook shape on their sperm head. So we measured the sperm head shape, and then we looked at things such as testis histology as well. And you can see here is in the middle is an example of a healthy pure subspecies male. And so this is a section of testis, and the little lines here in the middle are sperm that are about to be released. And so this is where the sperm are being produced. And you don't need to know that much about testis histology to be able to tell that this one on the right looks a lot different, and there's no sperm being produced, and this one is probably 100% sterile because I don't see anything being produced there. So that one's particularly bad. So we found that overall 30% of hybrid males had evidence for some reduced fertility. So they had testis weight and or sperm count that was outside the range that you see in pure subspecies. And it's important to note here that even though we see this high prevalence of problems, it looks like there's problems, we didn't directly test the fertility of these males. And so we didn't breed them to females. And most of the males that I brought back from the hybrid zone did produce offspring. So this is a case of subfertility in these hybrids, in most of them, rather than complete sterility, which is sometimes seen in the laboratory. And that's important because a few slides ago, I showed you about these different kinds of barriers that can happen in the hybrid zone. So it seems like, my hypothesis is that there's several different kinds of problems that are happening in hybrids, so that, you know, they do okay where they are. There's plenty of hybrids there, but they just have enough problems that they can't escape the hybrid zone and outcompete their the pure subspecies individuals. And this hybrid sterility is one of those problems. So now that we had an idea of what's going on with at the phenotype level, the traits, then the next thing we wanted to do was trying to find the genetic basis of these differences. So we can take advantage of the hundreds of generations of intercrossing and backcrossing that's happened in the hybrid zone to mix up the genomes between the domesticus parent and the musculus parent. So in this cartoon version here, they're shown in blue and yellow, and the hybrids that we catch are very late generation. So you see this mixture of different size regions from domesticus and musculus. And this is very advantageous for us because this mixing up is what we need in order to map the regions of the genome associated with the traits we see. So what we want to do is a genome-wide association study where we have genetic markers all throughout the genome, and we look for regions of the genome that are more similar between sterile mice and fertile mice. So as you can see here in this cartoon area, suggesting that whatever the allele is here, that's the domesticus allele, the blue allele, would be then associated with sterility. So just a cartoon version of what we would expect from each of those. So then, you know, each one of the markers here is a single nucleotide polymorphism. So one, you know, base pair change in the DNA. And so what we look for is basically march through the genome, and for each of those, look for if they have a significant association with our trait. So here's an example where you would show testis weight, where individuals that have 2 Cs at this position have higher testis weight than individuals with 2 Ts there. So that's what we're looking for. And the traits that we focused on for mapping were 2 traits: relative testis weight, which is testis weight normalized by body weight, and then this broad pattern of gene expression in the testis, so we looked at expression of about 30,000 genes, and then principal component 1, or a summary of this big change in gene expression that we see in sterile males. And so this is an example of the results for relative testis weight, where here the position in the genome along the 19 autosomes in the X chromosome is on the x-axis, and the y-axis is the probability of association of each little single nucleotide polymorphism with the trait. And so the ones that are above this dashed line there are significantly associated with testis weight. So we took those SNPs that were nearby each other and collapsed them into regions. And here's a summary of what we found throughout the genome for the 2 traits. So the regions in green on the left sides of the chromosomes here are the regions that are significant for testis weight. And the ones in purple, there's 60 of those, are ones that are associated with genome-wide gene expression pattern in testis. So what you can see is that there are a lot of loci that are associated with these 2 sterility phenotypes. They're found throughout the genome on every chromosome except for chromosome 18 for some reason. And so this is certainly an underestimate of the number of genes that are contributing to hybrid sterility, because we wouldn't be able to detect them all. And then that is not the only type of barrier there is. I mentioned the other type. So even in these early stages of speciation, you can see that there's a whole bunch of different genes that are contributing to this reproductive barrier. So this was a successful approach. It was the first genome-wide association study of a hybrid defect in nature. We had high mapping resolution. So if you cross the 2 subspecies in the laboratory to map these traits, then the regions you identify after 2 generations are about 30 megabases, whereas the size that we had was 2 megabases, so a lot smaller. So there are fewer candidate genes within those regions to help us figure out which are the causative ones. And more than half of the regions we identified had 10 or fewer genes, which is a reasonable number that we can consider each one of them individually. And many of these have previously known roles either in reproduction or in gene regulation, so represent promising candidate genes. So those are the individual loci that we identified. But I mentioned in the beginning that we expect that hybrid dysfunction is caused by interactions between genes, these Dobzhansky-Muller incompatibilities. And so because we have this model, typically looking for gene interactions with in this genome modification study, for example, in human health, is really, really difficult just because there's so many different possible interactions. But because we have this model, we know that each of those individual loci we found must have at least one interaction partner in the genome that's causing these problems. So we looked for those specific interactions between the individual loci we found. And here's an example of one where there's a region on chromosome 17 and a region on chromosome X, which interact with each other. And if you have the D here stands for domesticus and M for musculus. So if you have one or two copies of the musculus allele on 17, and you have the domesticus allele at this position on the X chromosome, then you have lower testis weight than the other genotypes. So you need both of those genotypes to have this interaction. So using this approach, we identified a large number of these putative Dobzhansky-Muller incompatibilities. Here you see the genome is shown in a circle from chromosome 1 through 19 and then chromosome X. And on the left are the interactions we identified for testis weight, and on the right for gene expression. And so you can see that there's a lot of these interactions happening. And some of the patterns we picked out were things that were previously known from laboratory process, such as the importance of the X chromosome, which is the general pattern in speciation. And but we also found other regions like here on chromosome 12, where we didn't have any previous evidence that this region was involved in sterility. And we also noticed some general patterns such as we knew that each locus had to have at least one interaction partner, but we actually found that each locus tended to have multiple interaction partners. So you can see this region on the X here interacts with many regions all over the genome. So there's this complex network of regions in the genome that are causing sterility. And then if we looked in a little bit more detail at some of these regions, such as this region here on chromosome 12, which interacts with the X chromosome. The region is 4.3 megabases. In previous studies with a different set of mice, but from the same region of the hybrid zone, they showed that both of these regions have reduced gene flow through the hybrid zone, so they have those sharp climbs like I showed as examples in the beginning. So this is additional evidence that they are contributing to reproductive isolation. And this region only has 12 genes in it, one of which has known role in reproduction and two of which have known roles in gene regulation. And one of these, RL4A, is a particularly good candidate because knockout mice, the mice that have this gene eliminated, have low testis weight and low sperm count, but are not completely infertile. So the phenotype of this knockout mice looks like what we see in hybrids, and so this seems to be a really good candidate gene. And it has differential expression between the two pure subspecies and between sterile and fertile F1s produced in the laboratory. So we have some pretty nice candidate genes to look at. And so I'll tell you why the next part of the story is a little bit more complicated. So that was our previous work. And now I'm going to tell you about why we have developed this new approach using wild-derived inbred strains. So that's the background on the hybrid male sterility side. And now I'll tell you about our background on the gut microbiome. So all of this work, all the new stuff is in collaboration with John Bain's laboratory, and he's at the Max Planck for Evolutionary Biology and at Kiel University. And so His group does a lot of different things looking at different aspects of microbiome, and we have focused so far on the gut microbiome. So as I'm sure you're aware, there are many different factors that affect what the bacteria are in our gut. So our diet, our lifestyle, our genetic environment, and the bacteria that are in our gut can have a lot of effects on our health. So for example, our immune function associated with different disease. And I think quite interesting is the recent work on gut-brain axis, so things that are happening in our gut that affect things happening in our brain. And the variety of bacteria that we see in the gut is they're very varied across individuals. So you can think of the measure of all the bacteria in your gut as being something like a fingerprint. So that is unique in some extent in a particular individual. So we are interested in seeing if having hybrid incompatibilities affects the gut microbiome. And this work was Jun Wang, who was a PhD student at the time in John's lab. And he looked at gut microbiota in wild-caught mice from the hybrid zone from the same region where I caught them. And he also performed a cross. And so for the wild ones, this is a summary of the data showing that you can see that the musculus individuals and domestic individuals here are filled in. And that's a summary of how they are different from each other in terms of gut microbiome. And then the hybrids here are in a separate position. So they're not intermediate between the two parents, but they actually have this quite different gut microbiome from what you expect in the pure subspecies. And this altered microbiome is also associated with changes in the gut histology. So they have a higher score of pathology, so higher inflammation, for example, than what you see in the pure subspecies. So this suggests that this change in the microbiome that you see in the hybrids is associated with bad health consequences. So that's what Jun observed in the wild hybrids. And then he also did a cross between two wild-derived strains, so a pure strain of domesticus and a pure strain of musculus, and bred them for 2 generations to do an F2 cross. And so like I identified those regions of the genome associated with sterility, he identified many regions of the genome shown here in different colors for different bacterial taxa. So regions of the mouse genome that affect these, which bacteria they have in their gut. So now we have candidate genes from my sterility approach and candidate regions from the mapping and the F2s. But how do we actually go after, figuring out which underlying genes are involved and how are they functionally causing these effects that we see? So that's a major challenge if you're working in wild mice, because each individual is unique, right? They each have a unique combination of genome. So testing the effects of something in a repeatable way is not really possible. And the power of using laboratory strains of mice is that they are inbred strains. So after many generations of inbreeding, each mouse from the same strain is basically a genomic duplicate of the others, right? So because of this, you can have biological replicates. So you can look at the same thing in multiple individuals. You can look at across at different developmental stages, and you can perform multiple different experiments using individuals with the same genomic background. So this really gives a lot of power to figure out exactly how a gene can affect a trait. So in order to harness this power, but still be able to study these regions that we've identified from nature, I bred a panel of inbred strains from hybrid zone mice. So this is my little picture of spotted mice so that we can have the same repeatability and look at different ages. So how we did that was established small breeding stocks from 4 different sites in the hybrid zone, 2 of which are more domesticus, so about 30 to 40% musculus, and 2 sites which are higher on the musculus side, so about 70 to 75% musculus. So we had little breeding stocks from those for 8 generations, and then started making inbred strains at that point by brother-sister mating. And so the definition of an inbred strain is at least 20 generations of brother-sister mating. And by that time, you have that where they're basically all identical to each other. So when you do this, you have a lot of problems because of recessive alleles. So I think I started with over 60 strains, and in the end got 8 of them to use for further purposes. And here's an example of what the genotypes could look like from these different ones. So here, domesticus ancestry is shown in purple and musculus is in green. And these are examples of the first generation males. And you can see, like in the cartoon version I showed you before, that the regions of different ancestry are quite mixed up with each other, and they're different sizes. And so with the idea of having these this panel of strains is so that we can look at things like this region here, say, which is domesticus in multiple of these strains, but musculus in others. And the that particular vertical slice is associated with different genomic backgrounds, right? So what's going on elsewhere is different. So we have the repeatability and as well as the variability here. So we've made 8 hybrid strains, as I said, 2 from each of the 4 sites. And for the next step, to be able to map loci in that population, we performed a G2 cross, so crossed them for 2 generations. And so for each one of the, it's a little bit complicated, so bear with me here. So we took one more domesticus mouse and one more musculus mouse, and then a separate more musculus mouse and more domesticus mouse, and crossed those for the first generation to produce these G1 mice, and then crossed different G1s to produce G2s. And each G2 has 1 grandparent, so shown here in the 4 colors, from each of the 4 sites, right? So, and we did that in 8 different combinations. So this is taking these admixed genomes that come from nature and then mixing them up in different combinations so we can find which regions of the genome are associated with the trait. And yeah, like I mentioned, each G2 has 1 grandparent from each site. And we did this in 8 different combinations so that, and then about 40 males for each of the subclasses here, so that our sample size in the end was 320 mice. And this was coordinated, the initial genotyping and phenotyping was coordinated by Shawnee Doms, who's a PhD student in John Baines's lab. And so we raised all those males to 13 weeks, and then we genotyped them using this mouse genotyping array that has 140,000 SNPs. So another benefit of working on mice, there are existing genotyping arrays out there. And we looked at a whole bunch of phenotypes. So I'm going to tell you today about the gut microbiota and the fertility phenotypes, but we also measured them and got samples for lung microbiome and things so that we can get as much as possible as we can out of the same set of mice. So I'll tell you about the gut microbiome mapping, which is, well, it's being written up right now, so it's further along, and then the sterility mapping, which is a little bit earlier. So Shawnee has been doing the gut microbiome mapping, and you know, like I mentioned before, we're interested in what are the genetic determinants of the microbiome and how this affects the health of the mice. So interested in how the genotype affects the individual variation in microbiome, and then how that could potentially contribute to speciation. So here's a phylogeny showing the 2 subspecies and within their broader phylogenetic context. So to measure the microbiome, we measured this gene 16S ribosomal RNA from the DNA and the RNA in the gut. And the 16S is a common method for profiling microbiome communities. And the DNA measures the standing community, so basically measuring the number of cells of different microbiota. And the RNA is an estimate of the active community, so which ones are producing lots of proteins, are really active in the gut. There's many different taxa that are present if you look across all the mice. So we focus on the core microbiome. This plot is showing the abundance within a mouse and or the average abundance across mice, and the y-axis is showing the how many mice it's present in. And so we looked at the core microbiome, which are those taxa which are present in at least 25% of the individuals, and their median abundance is greater than 0.2%. And this slide shows the variation in the core microbiome across the 8 subclasses for DNA and RNA. And the point here is just to show that they're quite variable, right? So there's a lot of variation happening in there at both the DNA and the RNA levels. And so we know that there's a lot of variation, but how much of that is actually heritable variation? And so we're estimating that here for the DNA and RNA. And what's shown in green is the genetic variation associated with individuals. So the things that we can map. And so for some things like this bacteroidetes, it's very high, over 90%. And then some have no detectable heritable variation. So for those, we cannot find regions of the genome associated with it. That the variation among the individuals there is residual, so environmental. And then what's shown in orange there is the mating pair. So basically, how much of the variation is due to the fact that they had the same parents and were in the same cage, different from their genetics. So quite a few of these traits we have good power to be able to detect loci. And then this is showing the regions of the genome. So here, now the chromosomes are shown as horizontal bars from 1 through 19 and then the X. And this little heat map shows how many different bacterial taxa had significant associations with that region of the genome. So you can see that there's quite a few significant associations there, and here they are broken down by taxonomic level. And ASV here is sort of equivalent to a species. That's a sequence variant is what that means. With microbiota, you can't really do the biological species concept, so you have to do things by sequence similarity. And so, yeah, so we found a lot of regions of the genome that are associated with bacterial taxa. Some of them, for example, this region of chromosome 7, there's a large region that's associated with a large number of bacterial traits. So this seems to be a hot spot in the genome for affecting microbiome variation. There's other regions, such as this one here on chromosome 17, where there's a lot of taxa affected by a smaller region. So there we might be able to more easily pick out what are the underlying genes. And overall, if we look at individual loci, the median size here is a little under 3 megabases. So I mentioned earlier that if we had done a similar number of generations of crossing using pure subspecies, it would probably be about an order of magnitude bigger, so about 30 megabases. So by crossing these hybrids, we have a lot better possibility to narrow down the regions. So now we have all these loci. How do we figure out what candidate genes we want to look at? So one thing we did was make this string network here, and this is looking at the genes that are closest to each significant SNP in the genome, and then looking to see if they have previous evidence from published data that those proteins interact with each other. And so the lines here are showing genes that are associated with bacterial taxa that have evidence from mice that they interact with each other. And you can see that there's a lot of connections here. This is highly significant. And so we might want to look, for example, at regions of the genome like this one here, or not regions of the genome, but sets of genes that are known to interact with each other and also that we find significant associations for. And then in a little bit more detail, we can also compare the candidate genes we have to previously publish research for related questions. So there was a paper earlier this year that looked at gene expression in different organs in germ-free mice, so that are born without a microbiome, and conventional lab mice. And so they found 216 genes that were significantly or highly ranked associated with the colon. And out of those 216 genes, 169 of them are in our region, so are significantly associated with microbiota in our mice. So that's encouraging, so suggesting that we're finding things that make sense. And one of the candidate genes I'll tell you about is this one, SLC26A3. And this is a chloride anion exchanger. And so this is a blow up of one of the significant regions. So you can see here is the gene, and the little pink dots are the significant SNPs. And this particular gene is associated with multiple bacteriotaxa, so 4 ASCs, or like species, 2 genera. And it's, as I mentioned, it's chloride anion exchanger. It's expressed in the intestine, and it's involved in resorption of liquid from the intestine. And in that the study I just mentioned, they found that it's overexpressed in germ-free mice in comparison to these conventional lab mice. And it also, there's the knockout model for this gene has diarrhea and altered gut microbiome. And the gene has been associated with diarrhea in humans and human ulcerative colitis. So there's a lot of evidence that this gene plays an important role in gut microbiome and has health effects. So that seems to be a very good candidate gene. And so then the next step with these candidate genes will be to functionally characterize them in the hybrid strains that we used in the cross. So we can go back to the same alleles that we found in the association in this cross, and then look at them in this repeatable way. So that is a summary of the gut microbiome. And then the sterility traits, a very similar approach. My PhD student in Bath, Pagan Aspinall, has been doing that part. And so the first generation mapping that I told you about before, we looked at testis weight and this overall gene expression, which are kind of gross level phenotypes, big phenotypes. But these are different examples of histology in those males. So this one, A, is the normal one, and all the other ones are hybrids that have problems. And you can, if you look in detail, there different hybrids have different defects. So there's a variety of different things that are causing these problems. So for example, this male here has low sperm count and also low testis weight, whereas the male here in H had low sperm count and normal testis weight. So there's more going on than what we can measure with just these gross level phenotypes. Spermatogenesis is a complicated process that takes 35 days for mitosis, meiosis, and spermiogenesis to occur. And this is a little map of what you see in one section of testis. And here's an example where you see the cells go from mitosis on the outside of germ cells, and then meiosis, and as you move towards the center, going through this process from outside to inside until they release the sperm. So, and here's a cartoon version of how you have the mitosis, meiosis, and then round spermatids and sperm. So one thing that we can do is look a little bit more at those different cell types. And so Pagan did histological sections of testis, and then this is a whole section of testis, so with many different tubules. And she looked at each of these and counted the different cell types. So how many spermatocytes, round spermatids, et cetera, there are. And did things like measuring how the area of the tubule and things like that. And then she also counted particular defects that she saw. So for example, this one is showing degeneration of the epithelium. This one is showing vacuolization, exfoliation, so these different defects. And so she measured a whole bunch of different phenotypes. So gross ones, fine ones, and these defects. And then we mapped them using the same approach, the same genotypes. And this is a summary of significant SNPs at The ones in blue are very, very highly significant, and the ones in pink are intermediate level. So these are SNPs associated with different phenotypes. And like I mentioned, these are very recent results, so we're just starting to get into them in more detail. Here's an example of one which is for the number of spermatocytes. So those are the cells that undergo meiosis. And so counting how many of them there are in each tubule, and you can see that there's one region on chromosome 6, which has a big region associated, and then several other loci. So now we're at the point where we're looking in there at candidate genes, and then the next step there will also be functional characterization. So overall, this approach is to take advantage of these hybrid zone inbred strains so that we can cross them again in new combinations and combine genotype and phenotype to try and understand what is the genetic basis of these hybrid dysfunction traits. And so we're now at the point where we have good candidate genes for gut microbiome and working on it for sterility loci. And as I mentioned, the next step will be to functionally characterize those within the hybrid zone strains. And so to go back to the big picture of understanding speciation, I mentioned in the beginning that mice are a really good model because they're in the early stages and because we have all these genetic tools. So I see mice as sort of a gold standard where we can test these associations between genotype and phenotype and ultimately fitness with a lot of precision in the laboratory. And then we can use the results that we find to translate to other taxa that hybridize in nature. So here's a nice panel of different organisms that some of which cannot be crossed in the laboratory, but we can still learn a lot using similar approaches. So thank you very much for listening to my talk. And I want to acknowledge my co-authors here, and then funding sources, and the mice, of course. So here's one of my hybrid mice, and it's sitting on a beer stein because they're from Bavaria, of course, which is very important for beer. So thank you.
Thank you very much, Leslie. That was an excellent talk. So we have some time for questions. So the first one is, what is it that caused the speciation of these three species of mice that you're studying?
Yeah, so I think I forgot to mention that when I showed the hybrid zone slide, that the mice came, they developed in like India type of area, we think, or and then there's also a lot of diversity in Iran. And then so that's where the subspecies first, or the species first arose. And then they moved through different pathways, these two, into Europe through two different pathways. So they had been separated for some time, and then they eventually met together again, and that's when they formed this hybrid zone. So they went into Europe through two different pathways. And so it was just this time as they were apart from each other. A lot of times people ask me if there's ecological differences between them, but they all, they're commensal, so living with humans, and so there's no clear difference in their diet or environment. It's just the fact that they were separated for some time and had time to accumulate these genetic changes.
Oh, okay. So their migration to Europe, is it recent?
No, that was, let's see, the hybrid zone. Yeah, I mean, I guess relatively recent. So probably about 10,000 years or something since they came back together.
Okay, so we have two questions that are related to the hybrids. So the first one is, why is it that these species of mice hybridize if their offspring will be sterile? And the second is, why is it that different hybrids seem to be affected in different ways?
Yeah, those are good questions. So yeah, so it doesn't seem like it's a very good idea for them to produce hybrids if the hybrids are going to have problems. But if you remember from when I showed the chart showing the change in the background of the mice over space, so it is only over 40 kilometers, but at least now the pure domesticus and the pure musculus don't actually meet each other. So it goes from 100% to 90%, and then very quickly from 70 to 30. But if they're going to be able to tell each other apart as mates, usually in mice they do that by smell mostly, then they can tell, a domesticus versus a musculus. But I'm not sure how well they can just tell, 100% domesticus to 90%, et cetera. And so we would expect that they might develop those C-Manning barriers. So this is through a process called reinforcement, where you have the post-zygotic barriers, so the problems in hybrids, and then there's selection on the individuals to stop mating with each other. But it's pretty hard for them to distinguish is the issue. And then the other issue is that, the reason that they can hybridize is because they do have, some problems, but none of them is strong enough. Like I mentioned, the sterility, that most of the males have some level of fertility. So you can still get gene flow happening there, and to actually get rid of it completely will take a long time. And actually, the other side of the coin that I'm not telling you about is, one of the things we're looking at is some genes move very quickly through the hybrid zone, so they can actually be beneficial. A gene that's beneficial would move quickly through. This isn't a particular example here, but one example from wild mice that moves around quickly is pesticide resistance, right? So if you have an allele to that in one area, it'll quickly move to other populations. That answered the questions. I'm not sure I remember both of them.
Yeah. Oh, yes, that why is it that different hybrids are affected in different ways?
Yeah, so different, so it's that different, it depends on which set of alleles they get. So, I showed that, that kind of complicated picture with all the different interactions between the loci. And so, well, my hypothesis at least is that the more of those Dobzhansky-Muller incompatibilities that you have, the worse you are in terms of your fertility or your gut microbiome. And there's many different ways that things can go wrong. So you could have the trait I showed kind of at the end, where the number of spermatocytes, so how much meiosis is going on. But there's also things in terms of the shape of the sperm, or how much it swims. So there's a lot of different places that can go wrong, and there's a lot of different genetic changes that have happened. So there's many different ways that you can mess it up, basically.
Okay, thank you. So another question on the hybrids is, do they cope better with environmental stress in those regions, or do they have any advantage?
Yeah, so the downside of working on house mice is because they're commensal, like I mentioned to answer to the previous question, there's not a clear environmental difference that's happening. But sometimes hybridization can be a positive, or I don't know about positive. It can open up new niches. So for example, a former colleague of mine, Arne Nolta from the Max Planck Institute, he works on codis, this sculpin fish that live in Germany also. And there's two different species that live in small streams. And then the hybrids between them can live in the main channel of the River Rhine. So they're able to be in that habitat that the two perennial species cannot. And so, that's only in the last few hundred years. There's also examples of that in sunflowers. And my former colleague, Rika Stelkens, who's now in Sweden, in Stockholm, I think. She looks at this in yeast hybrids and does hybrids in the laboratory and then tests them in a whole bunch of different conditions, and has shown that sometimes hybrids are able to deal better in different conditions. So this could potentially be interesting in the context of climate change, maybe not with yeast, but with other organisms, that where you could have more hybridization, because as species ranges change, there might be coming into contact where they didn't before, but not so much with mice, unfortunately.
Well, we actually have a related question to that. Can hybrids ever lead to forming new species?
Yeah, so that is, like I mentioned, the caudus species, and that's a case of homoploid hybrid speciation. So that's where there's not a change in the chromosome number. A more common kind of hybrid speciation is through ones that are where you keep one set of chromosomes from each parent. So it's an allopolyploid. So they have, so like most of the plants that we eat are actually hybrids, and plants hybridize a lot through this method. And yeah, so that's where you have multiple copies of each chromosome, and actually it's taken advantage of a lot in breeding. So the reason that bananas don't have seeds, which is nice for eating them, is because they're well, they're triploids actually, but they're hybrids, and so they're sterile hybrids. They don't produce seeds, and that's nice for eating.
Yeah, thank you. So we have two questions relating to the sort of the mechanism. So the first one is, are all the genome areas associated with these hybrid sterility related? Are they all related to reproduction?
No, well, I mean, so we don't, we haven't identified yet any like specific genes that we know are involved with either the gut microbiome or the sterility regions. The one gene that is known for hybrid sterility from other work in mice is PRDM9, which is a gene that is actually involved in the rate of recombination. And so I'm not sure if it was known that it was involved in fertility before, but it's not, you know, it wasn't clearly known to have a link there. And so there could be genes that are things that we know or have a role in sperm morphology and things like that. But there can be other ones like I mentioned the candidate gene we have, which is involved in, you know, ion exchange in the gut. There's another candidate from the gut microbiome that is a circadian rhythm gene. And so, you know, it affects the daily cycles, and that can also affect the gut. Yeah, so there's a lot of genes that can have kind of unpredictable effects on traits.
Yeah, thank you. So are there any similarities with hybrids from other species in terms of the testes abnormalities?
Yeah, so one of the things that I mentioned briefly was the role of the X chromosome, and that's because of this pattern called, described by Haldane, so Haldane's rule, which is that if you have hybrids that have problems, and there's a difference in the degree of the problem between males and females, then the sex that is heterogametic, so having two different sex chromosomes, will have problems first. So in mammals, mice, and Drosophila, there's, you know, XY males. And so males only have one copy of the X chromosome. So that means that any gene that's causing a problem on the X, the males only need one copy, whereas the females would need two copies of that to have a problem if it's interacting with something else. So the role of the X chromosome, and then in other species, it would be the W chromosome, or ZW species. Oh, no, the Z chromosome, sorry. So that's one common pattern. And then the pattern that I showed with one region having multiple interaction partners, there's some evidence from other taxa that that's also the case. Trying to think of other general patterns. That's all I can think of right now.
Okay. Are females also sterile?
Yeah, so there is one study, at least, showing that actually looking at this carefully in hybrid female mice, it's a lot harder to study because in males, you know, we can just count the sperm and look at the testis, whereas in females, meiosis occurs, the meiosis that leads to eggs happens before the mice are even born, also with us, right? So it's very difficult to study. but also because of Haldane's rules and evidence from laboratory crosses, we know that it's not as severe in females as it is in males. So we've looked less at it, but there certainly is some reduced fertility. I also saw that anecdotally just in the mice that I brought back from the hybrid zone, some females just never had offspring.
Okay. So we have a question on the implications for humans. So were hybrids from Neanderthals and humans also sterile? And have we inherited any sterility genes from mixing with them?
So that's a good one. I mean, they were at least not completely sterile because we know that there was some introgression or movement from genes from Neanderthals to humans and vice versa, as well as with Denisovans, the other ancient species that's been found. The degree to which they were had reproductive incompatibilities is not well known. My guess would be not that much because the genetic divergence isn't that high. And then I believe, I don't actually know that much about this, but I believe there are fewer loci on the X chromosome than is expected, which is what you would, that have moved between. So that would suggest that, and that genes that cause problems with at least, male fertility would not have moved through as well. But there are certainly, so PRDM9, which I mentioned, is the one known sterility gene in mice. So it's also important, it has the same function in humans as recombination. And within humans, there are different alleles of PRDM9 that are associated with problems with fertility. So even though we talk about these hybrid incompatibilities from interactions between genes, you can expect that sometimes you can have effects of interactions between proteins from a male and a female within humans that can be a problem. So for example, a couple where there's no evidence for fertility of the man or the woman, but they can't have kids together for some reason.
Okay. So we also have a few questions on lab mice. So why is it better to study wild mice instead of lab mice?
Yeah, so that's a good question. I probably should have included that. So yeah, so lab mice are, I like to say it's like studying dogs versus wolves. So lab mice are developed from pets. People had mice as pets. And so for one thing, they've been like domesticated. Basically, they're very tame. So I always have to warn people who take care of my mice in the mouse facilities that my mice, you open the cage and they just jump out and sometimes they bite and things like that. So they're much wilder. And they also have a lot more genetic variation. So it's also similar. And well, dogs appear to be more variable because their traits vary so much. But actually, if you look at wolves, the amount of variability, genetic variability that's there is much higher. And that's also true with the wild mice. And then on top of that, I'm particularly interested in finding out with what is causing speciation in the wild. So looking in this natural hybrid zone to figure out, which are the regions of the genome that are actually keeping those mice as separate subspecies right now, in the wild. So that's why I'm interested in them. But aside from, the interest in speciation, there's been a growing interest in looking in, well, at least getting some variation from the wild and then studying it in the context of biomedical research too, because of this greater variability. And for things like immunity, for example, like wild mice are much more similar to humans, than a lab mouse would be after all that selection. There's benefits in that way too.
Okay, so the next question on lab mice are different lab mice strains different species, and are their hybrids also sterile?
Yeah, so there's the traditional, what we call the traditional classical lab strains are pretty much all from one of the subspecies I talked about, which is Mus musculus domesticus. So that's, I talked about the hybrid zone, and that's all through natural range expansion. But the mice that domesticus has been brought by ships and stuff all over the world. So like in the Americas, there's house mice, but those all came from Europe. Those are all domesticus. And like in Australia and stuff like that, those are all domesticus. They're all over the place. And so those are almost all the lab strains are domesticus, or all of the classical lab strains are pretty much all domesticus. There are some wild-derived inbred strains. So the same way I took mice from the hybrid zone and then crossed them mice from the same place. So, you know, we know exactly where they came from. There are also wild-derived strains of mice, but those are separate and haven't been traditionally used as lab strains from each of the subspecies. And yeah, so then if you cross those, like from musculus to the classical lab strains, sometimes they're sterile, but not between the lab strains. I'm not sure if I know of any. I don't think there's any of the classical strains that you cross have fertility problems, because they're all domesticus, yeah, so they should be all right.
Okay, we also have a question that's asking, why is it that lab mice are white and house mice are brown?
Well, there are actually a lot of, we think of lab mice as being white, but actually the most common one, and I think the one I used with it has a little DNA tail, black 6, so the one that had the first genome is actually black, hence the name black 6. The color that's shown in the picture of mice that's in my talk, the brown color, that's called a Judy, that is the natural one. But in the same way that, you know, wolves are all kind of the same color, but then when you breed dogs, you can get all the different colors. That's the same thing. So that the lab strains, one of the things that they were bred for was different colors. So they're called fancy mice. So before they were used for research, there were people that would breed them to try and get these different colors. And so they're naturally occurring variants there, but that get selected for. So like, well, my favorite mouse from the hybrid zone was this really pretty light gray color. And so that's a natural variant, but most of them look this brown color. Oh, you're muted, Araxi.
Yes, I just realized that. So we have a question on the microbiome. And the question is whether the microbiome of the hybrids looks like an intermediate of the two parents, or does it look like a stressed, diseased microbiome?
Yeah, so it doesn't look directly intermediate, so it does look different. And, you know, at least with the ones that we, from the wild, you know, we associate it with problems like inflammation, so things that look like disease. But there's some things, like if you look at particular bacterial taxa, some of them are in between. So it's kind of a mixture between things that are in between the two parental species. So probably just an intermediate phenotype that doesn't necessarily do anything bad, and some of them that look much worse, you know, that have an altered microbiome that probably has negative effects.
Okay, and I think we're going to close the questions. The last question we have is also on the microbiome, and that is, are people from different cultures and with different diets ever incompatible with each other because of their
that's
microbiomes?
A good question. Yeah, I don't think that they would have any you know, speciation type issues. And within humans, we have, you know, there's no evidence for reproductive barriers between, you know, people from different populations. There's, you know, a lot of variation that is shared among humans. So there's no evidence for that. But certainly humans do have varying gut microbiomes based on their diet. And you know, that can have effects on it. And there can certainly also be genotype by environment effects, right? So if you have two different people eating the same diet, you know, one of them can have a better microbiome than the other. So there's still genotype by environment effects, not related, I don't think, to the mixing of populations.
Well, I think that's all. Leslie, thank you very much again for giving this excellent talk. I think we've all learned a lot about speciation, and maybe sometimes we underestimate the value of house mice. We often see them as pests, but they are also an opportunity to uncover basic principles about where do we come from and where do new species come from. So thanks a lot for opening our eyes to that. And so I also want to thank Colegio Nacional and Fundación UNAM for their support to the Universities for Science Consortium, and also, of course, to every single institution that forms part of the consortium that includes the Milner Center for Evolution and the University of Bath. So Leslie is not only closing this series, but also she's the fourth academic contributing a talk to this series from the Milner Center. So we appreciate obviously the support and also the fact that they are part of this consortium. And obviously we also appreciate the support from all the other institutions that are part of the consortium as well. So although the series is closing, so the seminar series of Universities for Science Consortium is closing with this talk. We have a special event, and that will be the last in the year, and that will take place on the 10th of December. So to give details of that, because the talk is going to be in Spanish, so I'm going to switch to Spanish. So los invitamos a todos al último evento que tendrá lugar el día 10 de diciembre a las 12. Es una plática especial, una plática plenaria por el doctor Carlos Frank de la Universidad de Durham en el Reino Unido, y él acaba de ser galardonado, tiene una carrera estelar y acaba de recibir la medalla y el premio Paul Dirac del año 2020, y nos